Hello, my name is Professor Hudson. You are about to view a narrated PowerPoint lecture that focuses on how cell division is controlled. Let us begin. The activities of specific proteins drive the cell cycle. During development, after an injury, or to compensate for normal wear and tear, a variety of cells release growth factors, which are hormone-like molecules. The cell cycle is driven by proteins called cyclin-dependent kinases, or CDKs. In order to stimulate cell division, growth factors must bind to cell surface receptors, leading to cyclin synthesis. Then the cyclins bind to and activate specific CDK molecules. The cyclin-CDK complex then stimulates synthesis and activity of proteins that are required for DNA synthesis, causing the cell to enter S phase. Other CDKs, which stands for cyclin-dependent kinase, become activated during G2 and mitosis and promote events in those phases, such as chromosome condensation, nuclear membrane breakdown, spindle formation, and attachment of the chromosome to the spindle. Still, other CDKs stimulate processes that allow the sister chromatids to separate into individual chromosomes and move to opposite poles of the cell during interphase. Although CDKs drive the cell cycle, multiple checkpoints ensure that the cell successfully completes DNA synthesis during interphase. Proper chromosome movements occur during mitotic cell division. So growth factors bind to receptors. They stimulate the production of cyclins, and then cyclins bind to and activate CDKs, which then start the process of cell division. There are three major checkpoints in the eukaryotic cell cycle, each regulated by protein complexes. One is at the junction of G1 to S. Basically, the cell needs to check to make sure that the DNA is intact and suitable for replication. Then there's another checkpoint at the junction of G2 to mitosis. The cell needs to check to make sure the DNA has been completely and accurately replicated. Then there's a third checkpoint at the junction between metaphase and anaphase. Are all of the chromosomes attached to the spindle and aligned at the equator? If the cell fails any of these checkpoints, the cell is supposed to kill itself through a process called apoptosis. However, sometimes the cell does not, and that cell with its defects could go on to reproduce more cells with a defect, leading to the formation of tumors. The checkpoints ensure that cell division occurs as it should. When meiotic cell division doesn't occur as it should, cancer can result. Let us review. Question 11. You sprain your ankle. Platelets are attracted to the sprained area. Why do they release platelet-derived growth factor and epidermal growth factor, which activate the cell cycle? Choose the correct answer. Now let's talk about cancer. As mentioned previously, if the cell cycle does not occur the way that it's supposed to, cells can divide uncontrollably. Uncontrolled cell division is cancer. So let's take a closer look at cancer. Cancer is a mistake in the cell cycle. Cancer stems from a failure of the regulatory controls that govern cell division. As mentioned previously, they are checkpoints during the cell divisional cycle. If the cell fails these checkpoints and the cell does not kill itself through apoptosis, it could potentially become a tumor, which can lead to cancer. Let's talk about mutations. Mutations in DNA are the primary reason why a cell will fail these checkpoints during the cell cycle. A mutation is a heritable change in genetic material. A mutagen is something that alters or mutates DNA. Mutagens can be environmental, they can be X-rays, chemical, radiation, virus, or they can happen spontaneously. There are two main categories of mutations. There's germline mutations and somatic cell mutations. Germline mutations are mutations that happen very early in the formation of the embryo or in the sperm and the egg. Germline mutations will occur in every cell of this person's body, and they can pass them on to their offspring. Somatic mutations occur much later in development and typically only affect a particular area of the person's body. They do not affect the sperm and egg. Somatic cell mutations affect the individual but will not be passed to the offspring. So mutations in DNA can cause failure of the checkpoints in the cell cycle. Failure of these checkpoints can lead to uncontrolled cell division, which results in a tumor. This is the definition of cancer. Cancer is a disease caused by gene mutations. When cells initially mutate in certain genes, leading to uncontrolled cell division, two possible types of tumors can occur. A benign tumor, which is an abnormal mass of encapsulated cells and non-mobile, or a malignant tumor, an abnormal mass of non-encapsulated cells capable of metastasis. Benign tumors do not move from their original location, whereas with malignant tumors, the cells can break off and migrate to other parts of the body, setting up new tumors. Let's look at some features of cancer cells that make them different from normal cells. Cancer cells versus normal cells. Cancer cells are non-differentiated cells. They forget their identity. They do not specialize the way normal cells do. Cancer cells will have abnormal nuclei. Often, cancer cells will have nuclei that have way more chromosomes than what they're supposed to. Sometimes cancer cells will have 2 or 3 nuclei per cell, which they're not supposed to. Cancer cells do not undergo apoptosis. Cancer cells will not kill themselves and take one for the team like a normal cell will. Cancer cells lack contact inhibition. Cancer cells do not understand that they are supposed to stop dividing once they reach another cell. They will grow all on top of each other. This leads to disorganized, multi-layered sheets of cancer cells. And cancer cells have the ability to undergo metastasis, which means they can go to distant sites in the body. Normal cells have what we call addressing proteins on their surface, which help them know what part of the body they are supposed to be in. Cancer cells forget their address and can detach and go to anywhere in the body. Cancer cells do not behave like normal cells. Let's look at the development of cancer in more detail. All cancers start with a single cell that mutates, and the mutation causes repeated cell division. As new mutations arise and one cell now has turned into several cells with the mutation, these cells now have the ability to start a tumor. Then some additional mutations will occur. At some point, the number of mutations in the cell cycle genes will get to the point where the tumor now becomes invasive, where it can burrow down deeper into the tissue. And then at some point, additional mutations may cause this invasive tumor to turn malignant, which means it has the ability to invade the lymphatic and blood vessels. Once in the lymphatic and blood vessels, these cancer cells can migrate to any part of the body and set up new tumors. At this point, we say the cancer has metastasized. Cancer is often described in various stages. A malignant cancer has lost normal growth regulation. Invasive cancer can invade healthy tissue. And metastatic cancer can migrate to other parts of the body. Left untreated, malignant cells will cause the death of the organism. Let us review. Question 12. A disorganized, generally encapsulated mass that does not invade adjacent tissue is known as a. Choose the correct answer. Let's spend some time talking a little bit about the types of genes that have to be mutated to get cancer. While mutations can lead to cancer, the mutations have to be within certain genes in order to get a cancer. Therefore, not all mutations will lead to cancer. As mentioned, some cancers are caused by hereditary, which means they're inherited. Cancers can be caused by radiation, organic chemicals, viruses. For example, the human papillomavirus has been linked to cancer. And the human papillomavirus normally causes genital warts. As we mentioned before, normal cell division occurs through growth hormones that stimulate certain molecules that turn on or off the cell cycle. If you get mutations in these genes that regulate the cell cycle, you can get a tumor which can become cancerous. The two categories of genes that regulate cell division are the tumor-suppressor gene and the proto-oncogene. These are the two types of genes that must be mutated in order for a cancer to develop. Proto-oncogenes promote the cell cycle in various ways. If a proto-oncogene is mutated, it may become what we call an oncogene, and oncogenes cause cancer. Tumor suppressor genes are supposed to inhibit the cell cycle. If a tumor suppressor gene becomes inactive, it may promote cancer development. Normal cell division is a constant interplay between the products of the proto-oncogene telling the cell to divide and tumor suppressor genes telling the cell to stop dividing. Mutations in either one of these groups of genes can cause a cancer to develop. Think of it this way. The proto-oncogene is like the gas pedal or the accelerator in the car. The gas pedal and the accelerator makes the car go, just as the proto-oncogene makes the cell divide. Tumor suppressor genes act like the brake pedal in the car. The brake pedal makes the car stop, just like tumor suppressor genes tell the cell to stop dividing. If someone were to somehow put a brick on your accelerator, your car will continue to divide. Just as if proto-oncogenes are mutated, the cell will continue to divide. Tumor suppressor genes are in essence as if somebody cut your brake line. If you cut the brake line in the car will not be able to stop. Just like if the tumor suppressor genes are mutated, cell division does not stop. Proto-oncogenes act like the accelerator in the car, and tumor suppressor genes act like the brake pedal. Mutations of proto-oncogenes can have a lot of effects. The biggest effect is that the cell will not do apoptosis and kill itself, and it will continue to divide uncontrollably. Mutations in tumor suppressor genes can have various effects, but basically the cell will not do apoptosis like it's supposed to, and the cell will continue to divide. Some examples of tumor suppressor genes are P53 and RB, which stands for retinoblastoma gene. These are two genes in your chromosomes that actually are supposed to inhibit cell division and help to prevent cancer. In the next segment, you will have some review questions which you need to jot down the answers for the PowerPoint lecture quiz, and we will continue our discussion of cancer.